Welcome, I'm Francesco Polazzo, a PhD student at the IMDEA Water Institute of Alcalá de Henares, close to Madrid in Spain, and I'm part of the ITA and Marie Curie project EcoRisk 2050. And today I'm going to be talking about the effects of multiple stressors on the dimensionality of ecological stability. The overall idea of the dimensionality of stability started with this paper by Donahue et al. From 2013, which unraveled the multidimensional nature of stability. According to their work, the dimensionality of stability requires the assessment of several ecological properties to be quantified. Such study brought up how different stability aspects can be strongly or poorly correlated between each other. When stability properties are strongly correlated, the overall dimensionality is squeezed, resembling a one-dimensional concept, and all the correlating metrics give similar information. On the other hand, when stability properties are not correlated, the dimensionality increases, meaning that every single metric provides a unique information on the system's stability, and that every property should be considered for a proper ecosystem management. The multidimensional conception of stability is now becoming more popular, and today you can find some papers addressing the stability using its multidimensional conception. However, this is not a common practice in ecology yet. As shown in this chart taken from the review of Donahue et al., more than 90% of the study address stability using a one-dimensional approach. And they do that measuring stability in response to just one disturbance factor at a time. To expand our understanding of how multidimensional stability is affected by disturbances, we designed a freshwater mesocosm experiment in which we manipulated 3 common agricultural stressors: an insecticide, an herbicide, and nutrients. We applied the pesticides as a single pulse. On the other hand, nutrients were applied constantly throughout the experimental period as press stressors. In the experimental timeline, you can see that we sampled several times before and after the pesticides application, and that nutrients application started 4 weeks before the application of the pesticides. This allowed us to get to the pesticide application with our mesocosms in 2 very different situations. Half of them were in an oligotrophic status, where the others were in an eutrophic status. Oligotrophic mesocosms were characterized by higher biodiversity but lower biomass. On the other hand, eutrophic systems were characterized by lower biodiversity but higher biomass, and community composition was characterized by high dominance of tolerant species. As I said, we selected 2 pesticides as a model disturbances. Pesticides targeting different organism groups produce non-random reduction in diversity as well as in abundance of specific trophic levels depending on the toxic mode of action. Thus, pesticides can influence both compositional aspect of stability, such as species richness, and functional aspect of stability, such as abundance or biomass. We selected an herbicide to selectively reduce primary producers' diversity and abundance, and an insecticide to reduce consumers' diversity and abundance. Moreover, in complex communities, direct effects of specific disturbances are propagated throughout the food web, producing indirect effects. We then formulated some hypotheses on our experiment, and first of all, we expected that different disturbances will affect differently the dimensionality of stability, and as a result, also the correlation between the stability components. Secondly, we expected that the effects of the disturbances may be different depending on the trophic status of the system on which the disturbance were applied. Finally, we hypothesized that the correlation between composition and functional stability can be modified by disturbance. We sampled 3 organism groups: macroinvertebrate, zooplankton, and phytoplankton. From these samplings, we measured stability at the community and at the population level. At the community level, we distinguished between compositional and functional stability. Measurements of compositional stability were based on Wright-Curtis dissimilarity, and functional measurements of stability were based on abundance. At the population level, we selected 6 populations coming from different trophic levels. By doing so, each class of organism groups and their intrinsic sensitivity to specific disturbances were represented in the study. And we quantified multidimensional stability measuring 4 components of stability: resistance, resilience, recovery, and invariability. We measured the dimensionality of stability as the volume of the multidimensional ellipsoids constructed from the matrix of pairwise covariances between all the stability properties. To assess the effects of the treatment on dimensionality of stability, we used generalized linear mixed models, and we used the ellipsoid volume as a response variable. Stressors and their interactions were used as a fixed effect predictors. Then, to test whether the treatments affect the correlations between different stability properties, we first calculated correlation coefficients for each pair of stability properties within each treatment. We did that at the community and at the population level. And we use, again, generalized linear mixed model to test for significant effects of the treatment. Then we also tested whether disturbance can modify the correlation between functional and compositional aspects of stability. And to do that, we calculated the pairwise correlation coefficient between the same aspect of stability for function and composition. And also in this case, we use generalized linear mixed model to test for significant effects of the treatment. Let's move to the results of the study. And I want to start looking at the influence of the treatments on the dimensionality of stability. On the x-axis, you have here the treatments where I stands for insecticide, H for herbicide, and this is the pesticide mixtures. As you can see, at the community level for composition, we didn't find any significant difference between the treatments. But for functional stability at the community level and at the population level, we did find significant differences between the treatments. And I want you to notice that those differences were significant only in oligotrophic system. In eutrophic system, we didn't find any significant difference between the treatments. We then studied the effects of disturbance on the correlation between stability components. We found significant effects of disturbance on the correlation between stability properties, both at the community and at the population level. This means that different disturbances can modify the very nature of the correlation between stability components, which has important consequence for management. Finally, we tested the effects of disturbance on the correlation between the same aspect of compositional and functional stability. And we found significant effect on disturbance on those correlations. And this has even deeper management consequences, as we will discuss in a minute. Moreover, I want you to notice that most of the correlation were negative, both in eutrophic and oligotrophic systems, regardless of the disturbance type. Let's now discuss those findings. I want to start pointing out that our results show that functional stability was more affected by disturbance in oligotrophic systems. As oligotrophic systems were characterized by high diversity, the spontaneous question is somehow diversity involved in this finding? Well, the answer is, it depends. Indeed, we suggest that the response of functional stability to disturbance depends a lot on how you measure functional stability. Biomass and abundance do not account for disturbance-driven reduction in functional group. Thus, a community dominated by few but highly prolific tolerant species would result as more stable to disturbance compared to a more diverse community but composed by several sensitive species. This was the case in our experiment where species poor eutrophic systems resulted less affected by disturbance because of the total abundance that was still high. We argue that measuring stability, functional stability, using ecosystem processes as a common currency, and when I say ecosystem processes, I mean organic matter degradation, nutrients uptake, net ecosystem productivity instead of biomass, may lead to results very different from ours. Because measuring functional stability in this way would consider a reduction in functional groups, even if total abundance or biomass remain constant. Therefore, measuring functional stability in this way, only systems with higher or very high initial diversity may be able to maintain high functional level after disturbance application, as high diversity might ensure that different taxa perform the same function. Then the correlation between the same aspects of functional and compositional stability resulted to be mostly negative under different disturbances, and this has deep economic, ecological, and management implications. Indeed, managing systems for functional stability may have negative consequences for compositional stability, but also the other way around is true. This finding has profound implications for policymakers because they need to prioritize certain components of stability over others to meet relevant goals. For example, managing to improve only compositional stability, such as preserving species composition or diversity in a national park, will not necessarily improve functional stability and could have detrimental consequences for stability of biomass. Therefore, focusing either on functional or compositional stability in isolation risks an incomplete understanding of the effect of perturbation on ecosystems. And this is linked with a strong likelihood of underestimating the overall impacts. Finally, I would like to talk about the study's limitations and avenue for future research. We obtain our results performing a mesocosm experiment. Mesocosm experiments allow to test for different disturbances using a natural community composition. And also they allow to have a fairly number of replicates. However, mesocosms represent a simplified ecosystem in which some ecological dynamics may be limited for some organisms at least. Moreover, whether small spatial scale experiments such as mesocosms may be translated into general large-scale conservation policies has been questioned. Nevertheless, at present, mesocosm study represent the most reliable tool to test several ecological processes under peculiar disturbance regimes, such as pesticide, which cannot be tested in the nature for obvious ethical and environmental reasons. Secondly, which state variable is the most appropriate to quantify stability, especially functional stability, is still an open question. Indeed, functional stability is usually measured as biomass or abundance. We argue that measuring functional stability using several ecosystem processes as a state variable could be more exhaustive and would allow to study the relationship between multiple dimensions of compositional stability and ecosystem multifunctionality. Thus, our recommendation for future investigation is, first of all, not to use only distance-based metrics, but also to incorporate genetic diversity when studying compositional stability, and to link those measurements of compositional stability with multiple ecosystem processes, such as ecosystem respiration, chlorophyll A, nutrients uptake, and organic matter degradation. With that, I would like to conclude and to thank you all for your attention.